All right, it's time to get started. I'm going to say hi and welcome back to the next webinar in our Plantae Presents Global Plant Science Talk series. My name is Katie Rogers and I'm your host for today's webinar. This webinar series is brought to you by Plantae, the open online community for plant scientists powered by the American Society of Plant Biologists. I would like to give a special thank you to all of our ASPB members who are attending today. Your ASPB membership dues help support and make these webinars possible. For any of you who have not yet joined ASPB, you can join today and use the discount code PRESENTS10 to receive a 10% discount on registration. ASPB members get early access to these seminars. You can also learn more about ASPB and the opportunities we provide at aspb.org. Today's talk is the 17th Plantae Presents webinar in this series. If you haven't already done so, be sure to check out our archive and watch all of the previous talks in the series. You can find information on our blog and subscribe to our YouTube channel to get notifications when new recordings are posted. Before we get started, I'd like to go over a couple logistics. If you're having trouble connecting or need to leave the webinar early, just know that a recording of this webinar will be made available along with all of the associated materials within a few days. Today's talks will each be 20 minutes, followed by 10 minutes for questions. If you have questions for our panelists, please let us know using the Zoom questions and answer Q&A section. You'll also notice that there is a chat area open. That area is for you to introduce yourselves, say where you're from, and connect with your colleagues. Questions that are specifically for our panelists should be shared in the Q&A section to make them easier for our moderator to navigate. Matthias Benoit is here today to be our moderator. He is currently an HHMI postdoctoral fellow working in Zach Lipman's group at Cold Spring Harbor Laboratory in New York. His current and future research aims at understanding the significance of genomic and epigenetic variation in tomato development and breeding. Matthias is an assistant features editor at ASPB's journal, The Plant Cell, and is actively involved in mentoring students and promoting science communication through public outreach events such as this one. Thank you for joining us today, Matthias. I'm going to turn it over to you now to introduce our first speaker.
Thank you, Katie, and I would like to take this opportunity to welcome everyone to this new episode of Plantae Presents, whether you're watching it live or through the recording. I am delighted today to be moderating this session with two very exciting talks on the topic of plant epigenetics. So I would like to introduce our first speaker today, which is Claudia Köhler. So Claudia is a professor of molecular cell biology at the Department of Plant Biology at the Swedish University of Agricultural Science. She obtained a PhD at the University of Freiburg in Germany, and she's a member of EMBO, the Royal Swedish Academy of Science, as well as the German National Academy of Science, Leopoldina. So her lab has a broad interest in genetic and epigenetic mechanisms governing seed development and plant speciation. Her main focus is the endosperm, which is a tissue that supports embryo growth, similar to the role of the placenta in mammals. So research goals of her lab are to decipher the role of endosperm in controlling seed growth, regulating the embryo development, as well as establishing barriers towards interspecies and interploidy hybridization. Claudia, thanks for being with us today and looking forward to your talk.
Many thanks, Matthias. Many thanks for introducing me. Let me briefly share my screen. And I would also like to thank Mary and Jürgen for inviting me, and Katie for the logistics, and of course everyone for joining this seminar. What I would like to discuss with you today is basically depicted in the slide, which shows the enormous beauty and variety flowering plants have to offer, which ultimately gives rise to this question: how did this enormous variety evolve? And for good reasons, I put insects here in the central place because, without any doubts, insects had played a major role in plant speciation. But what I would like to convince you of, and I take the take-home message already at the very beginning, there are other, and in particular epigenetic mechanisms that also have a major role in driving plant speciation. These mechanisms act in the endosperm, and this is what this seminar will be about. Let me first ask the question, what is the key to speciation? Well, the key to speciation is to achieve reproductive isolation, which generally occurs by the accumulation of genetic differences over time, which finally cause two populations either not to be able to mate anymore, or if they are still able to mate, that the resulting progeny is inviable or sterile. And there are a multitude of reproductive barriers that either act before fertilization, and we term them prezygotic mechanisms, or after fertilization, and we term them postzygotic mechanisms. In both cases, they prevent the formation of a viable hybrid. Just to give you a few examples for prezygotic mechanisms, this can be, for instance, differences in flowering time or adaptation to different pollinators, or that pollen of one species is not successfully be able to germinate on the other species. If all those mechanisms do not apply, so if we have species that are still in principle able to mate, because they are still so closely related, but the progeny is inviable, these type of postzygotic mechanisms we are in particular interested in, because these are very early acting barriers. And one special case that I will mainly focus on in the beginning of my talk is when this species here has undergone, or this plant here has undergone, a polyploidization event, and as a consequence of it, it is reproductively isolated from this non-polyploid progenitor, which is not necessarily intuitive, because the species here has just double numbers of chromosomes, but otherwise it's genetically identical. Still, both species have a reproductive block. These triploid seeds are not viable, and the question we ask is, why is that? What is the genetic basis? Is this question relevant? Well, we think it is, because it provides one of the explanations how a newly formed polyploid plant can establish itself as a new species. Because polyploids arise very frequently in nature as a consequence of unreduced gamete formation, which is also a relatively frequent event. However, when a newly formed polyploid is formed, it is still surrounded by members of its non-polyploid progenitors. So in order for this newly formed polyploid to be able to form a new species, several things have to happen. First of all, it has to undergo a transition to selfing to ensure reproductive success. Secondly, it has to prevent continuous outbreeding with its non-polyploid progenitors, because pollen can still land, can still form inviable progeny. This process has to be prevented, and last but not least, it has to colonize ecological niches to prevent to be outcompeted by its non-polyploid progenitors. So these reproductive barriers have a very important role for a newly formed polyploid to form a new species. The question is, what is the genetic basis for these type of barriers? And as geneticists, we like to address these questions with genetic means, and for that reason, we are lucky that we have the ability to do so using our favorite model, which is in this case Arabidopsis. If you cross a diploid with a tetraploid Arabidopsis plant, in this case we use Columbia, because Columbia has a very strong block to these type of hybridizations, the resulting triploid seeds will look brown, shriveled, and the majority will be inviable. And as I said, we like to address questions with genetic means, and working with polyploids is not particularly easy, but we don't have to, because there are genetic mutants available, meiotic mutants, that have a meiotic failure, and as a consequence, they form diploid instead of haploid gametes. And this basically is this type of mutants are, for instance, Jason or OSD1. And these type of mutants allow us to mimic this type of scenario. So if we use these mutants as male pollen donors, we mimic this type of cross, we also end up with triploid seeds. So we can investigate this question using diploid plants, which, as you will see later on, has an advantage if one wants to do genetics. So what actually is the problem with these seeds? If we would look inside, what we would see is a small arrested embryo surrounded by a rather strangely looking endosperm. And if we look more closely using a confocal, what we would see, a wild type seed at around 6 days after pollination, the endosperm would start to cellularize. And this transition does not happen in a triploid seed. In the triploid seed, the endosperm remains uncellularized, and this actually causes the embryo to arrest for reasons we do not completely understand. So the main point so far is interploidy hybridization causes failure of endosperm cellularization, and these hybridization barriers are established in the endosperm. And because this talk will focus on the endosperm, just very briefly, how is the endosperm formed, to bring us all on the same page. The endosperm
is a product of a double fertilization event. It arises after fertilization of the diploid central cell it's fertilized by one of the sperm cells. Consequently, the endosperm itself is a polyploid tissue. It's a triploid tissue having 2 maternal and 1 paternal genome copies. The other fertilization event of the egg cell with the other haploid sperm cell will give rise to the diploid embryo. And the endosperm transfers nutrients to the developing embryo, so it's essential for normal embryo development. What now will happen is if instead of a haploid, a diploid sperm cell will fertilize a female gametophyte. Consequently, the endosperm will be tetraploid, the embryo will be triploid. And this tetraploid endosperm will undergo, or let's say will fail to undergo the developmental transition I just mentioned. It will fail to cellularize. As a consequence, this embryo will arrest its development and the seed will be inviable. And this phenomenon we term the triploid block. So basically it matters whether we have 1 paternal genome copy or 2 paternal genome copies present in the endosperm. The question is, why does it matter? And we can also rephrase this question and ask, how is increased chromosome number sensed in the endosperm? And over the years, a number of people in my lab have addressed this question. It started with 2 former PhD students, Philip and David, has been then followed up by 3 former postdocs, Hua, Gufang, and Herman, who all have their own labs now. It's currently followed or worked on by Chen Xing, who will also leave the lab end of the year to start his own lab in China. So, as I said, we wanted to address the question genetically, and many years back we started to do a genetic screen, a suppressor screen, looking for mutants that are able to form viable triploid seeds. And we identified a number of mutants that basically all shared the same phenotype. They allow to resume endosperm cellulization, and with that also the embryo could complete its development, forming a viable seed. So basically all our suppressors convert an inviable triploid seed to viable triploid seed. And over the years, we identified a range of suppressors, and I will not go much into detail, just very briefly. We can group them into 3 functional groups. Those 3 suppressors have something to do with heterochromatin formation. Those 2 suppressors something to do with small RNA formation or small RNA function, and PAC9 is the metabolic enzyme. So it looks rather like a diverse group of genes, but they share one thing in common, and this is their regulation. All our suppressor genes are regulated by genomic imprinting. And very briefly, what is genomic imprinting? Well, genomic imprinting is an epigenetic phenomenon causing parent of origin specific gene expression. And we can basically have 2 outcomes. Genomic imprinting can cause genes to be only expressed from the maternal alleles, and we term them maternally expressed genes, or in short MAGs, or it can cause genes to be only expressed from the paternal alleles, then we call them paternally expressed genes, or in short PAGs. And the specific phenomenon is driven by transposable elements that are generally found in the vicinity of imprinted genes. They recruit epigenetic modification and with that modify gene expression. And all our suppressor genes belong to this type of category. All suppressors are paternally expressed genes. With that, we ask the question, first of all, how is paternal specific gene expression established? And second of all, how does it connect to the triploid block? And what we and other groups already noticed many years back is that there's one particular epigenetic complex, the polycomb repressive complex 2, or in short PSC2, has a major role in this process. And very briefly, what are PSC2 complexes? These are epigenetic complexes modifying histones. To be precise, they modify histone H3 on lysine 27. They apply a trimethylation mark. And this trimethylation mark causes a change in chromatin structure and causes gene repression. And in plants, we have multiple variants of this complex, and one variant that is specifically active in the central cell, so the precursor cell of the endosperm, and later on also in the endosperm, is the FIS-PSC2. And Jordi, a former postdoc in my lab, asked what is the connection of FIS-PSC2 function and imprinting. And what he did is he profiled the presence of this modification in the endosperm, and he did that in an allele-specific manner. So basically distinguishing maternal and paternal alleles. And what he found is that the majority of PACs, so 90% of PACs, have a specific modification of K27 trimethylation present on the maternal alleles, while the active paternal allele is devoid of K27 trimethylation. So with that, the specific presence of K27 trimethylation established by the FISPRC2 is connected to genomic imprinting. And putting this into a model, how this comes about, what is likely happening is the FISPRC2 complex is, as I said, active in the central cell of the female gametophyte, so it will apply triisolation marks specifically on the maternal alleles in the central cell, causing repression. PRC2 function is not present in sperm, and in fact what has been recently published by Fred Berger's group is that there is specific histone variant present in sperm that cannot be targeted by PRC2 function, and with that the sperm genome will be devoid of K27 trimethylation, and with that the paternal genome has the potential to be activated after fertilization. And this parental specific expression is maintained after fertilization in the endosperm. So the FISPRC2 is not targeting the paternal allele after fertilization, and I will come back to that why most likely this is the case, because this is not necessarily intuitive. But the important point is PACs are regulated by FISPRC2 function. So how does that connect to the triploid block? Well, as I said, all our suppressors are imprinted genes, and all our suppressor genes, depicted here in red, are highly upregulated in triploid seeds. So the question is, why are PACs so particularly sensitive to genome dosage? Of course, in these type of seeds, we have double number of parental genomes, which nevertheless does not explain that we have an X-fold increase in their expression. So there must be something happening with those genes. And the answer to that question came when a former PhD student, Rita, together with Duarte, realized that the helitrons that are in the vicinity of imprinted genes are enriched for a particular sequence motif, which is characteristic for Metzbox binding for Metzbox genes. So this Carch box sequence motif is a characteristic transcription factor binding motifs for Metzbox proteins. And Rita was also able to identify the Metzbox protein recognizing these motifs, which is a type 1 Metzbox transcription factor Ferris1. So Metzbox transcription factors come in two flavors, type 1 and type 2, and most likely all Metzbox proteins you are probably aware of, like FLC, Gamos, Zipalata, Pistolata, etc., all belong to the type 2 Metzbox genes. Type 1, functionally, only very few are characterized. Most likely there's a high level of genetic redundancy because there are actually more type 1 than type 2 Metzbox genes, at least in Arabidopsis. So as I said, Ferris1 belongs to the less characterized type 1 Metzbox proteins. And the model that we now have is that because Ferris1 binds specifically to PACs, that Ferris1 is actually the reason why PACs are not targeted by the FISPRC2. In fact, it binds, the binding site on the maternal side is flanked by K27 trimethylation. So Ferris1 binds and most likely prevents FISPRC2 to act on the paternal alleles, causing the activation. And Ferris1 is probably also the explanation why PACs in general, and in particular also our suppressor genes, are highly upregulated in triploid seeds, because Ferris1 itself is highly upregulated for reasons we can only speculate on. But as I said, Ferris1 directly binds to PACs, including also suppressors, and increased dosage of Ferris1 will likely lead to increased dosage or increased expression of suppressor genes, including as well as all other PACs that are upregulated. If the scenario is true, as I showed you, these are suppressor genes. If we knock them out, we obtain viable triploid seeds. We would speculate that we should also obtain viable triploid seeds when knocking out Ferris1 function. So the question is Ferris1 indeed causally responsible for triploid seed arrest? And this is what we tested. So we generated Ferris1, Ferris2 double mutant, because Ferris2 is a close homolog to Ferris1. It's also physically very close, so we had to make a double mutant using CRISPR-Cas9. And when we look at these double mutant seeds, when they are triploid, they drastically differ from wild-type triploid seeds. So the majority of these triploid seeds are indeed viable. So Ferris1 is indeed causally responsible for triploid seed arrest. Well, so Ferris1 seems to be a major regulator of the triploid block, but it's not only that. What we actually found is that Ferris1 is a major regulator of a large battery of type 1 Metzbox transcription factors, as well as other transcription factors. And importantly, among those transcription factors is a list of genes that, yeah, well, I would term them the who's who of endosperm development. It's a number of genes that all have been shown to have a major role in endosperm development. And with that, we feel confident saying Ferris1 is a master regulator of endosperm development. Which of course gives rise to the question, how can a single gene become a major regulator of a whole set of genes important in one particular process, in this case, endosperm development? So the question that Rita, together with Yihun, addressed is, how did the Ferris1 controlled regulatory network evolve? And the answer to that question goes back to transposable elements, because what Rita found was that Ferris1 binding sites overlap with Helitrons, something what I mentioned already in the beginning. So, and Helitrons are also called RC Helitrons, which stands for rolling circle Helitrons, because during the transposition cycle, which involves a rolling circle intermediate, they take part of their flanking sequences with them and transpose them to different regions of the genome. And this is likely what has happened. So what we think is that an ancestral gene that has been under the control of Ferris1 has been targeted by a Helitrons. And during the transposition cycle of this Helitrons, it took part of the neighboring sequence, which in this case was including the binding site with it, and transferred it to different regions of the genome, bringing genes under the regulatory control of Ferris1. So which is in a way a very intuitive and easy mechanism, imaginable mechanism, how transposable elements generate transcription networks. And this concept is, I would say, pretty accepted in the animal field. It's not so much noticed yet in plants, but I think there are a lot of examples actually that point that this mechanism has been frequently used to generate transcription networks. And Yihun recently wrote a review on that I would like, or no, I would emphasize if you wanted to have a look at. Well, to summarize the first part of my talk, what I showed you is, or what I wanted to discuss with you is that changes in ploidy establish instant reproductive barriers in the endosperm. That secondly, imprinted genes act as speciation genes. That transposable elements can generate transcription networks and developmental novelties. And last, that increased dosage of the Metzbox transcription factor Ferris1 establishes the triploid block. In the second and shorter part of my seminar, I would like to give you a little insight into our work trying to understand the genetic basis of interspecies barriers. And our model system in order to understand interspecies hybridization barriers is the genus Capsella. And in particular, we are working on two species, which is Capsella grandiflora, an outbreeder, which you can easily visualize because it has large petals, and Capsella rubella, an inbreeder, as easily recognized by small petals. And both species rather recently diverged about 100,000 years back. And the work I will present you has started many years back by former postdoc Madame Caroline, then later on continued by Clermont, and is currently continued by Kasia and Laurian. What we notice is when we cross the inbreeder Capsella rubella with Capsella grandiflora, that the resulting seeds look phenotypically very similar to the seeds I just described before. So seeds derived from interploidy hybridizations in Arabidopsis. We have a arrested embryo, small arrested embryo, surrounded by a non-cellularized endosperm. And these seeds also look brown, shriveled, collapsed, very similar to interploidy seeds, suggesting hypothetically that maybe there's a common genetic basis underpinning this scenario. And what we also realized already supporting this view is that when we do a transcriptome analysis of hybrid seeds, that we have a large battery of type 1 Metzbox transcription factors, or AGLs, agamose-like transcription factors. So broadly speaking, homologs of Ferris1 deregulated in hybrid seeds. So in relation to both parents, if you compare to Capsella rubella, as well as if you compare to Capsella grandiflora. And importantly, in the parental species in Capsella rubella, all these genes have lower expression level compared to the outbreeder Capsella grandiflora. When we now ask, is this increased expression of AGLs functionally relevant? And we can only do a correlation, but this correlation looks like quite promising, I think, because when we tested whether those upregulated genes that we find upregulated in hybrid seeds, do they overlap with target genes of Ferris1 and Arabidopsis? So we determined the orthologs of Ferris1 targets in Capsella, and well, what we see is there's indeed a significant overlap, suggesting that this upregulation of type 1 Metzbox genes in Capsella hybrids has indeed functional consequences. And what we then did is that we asked where along the chromosomes those deregulated genes are located. And we found for something very striking, because instead of what one would expect, that they are somehow randomly localized along the 8 scaffolds that we have in Capsella, instead that they are highly predominantly present in those regions depicted in red, which are pericentromeric regions. So over all scaffolds, we have an enrichment of deregulated genes in pericentromeric regions, which we thought, well, is most likely not just by chance. Well, we know pericentromeric regions are heterochromatic regions enriched for transposable elements. So we asked, and these regions are highly condensed. If we now have a massive upregulation of deregulated genes in those regions, we asked, is there maybe a change in chromosome structure, chromosome condensation to be more precise? And this is what we addressed. What we did is we performed chromosome spreads from parental and hybrid endosperm, and what you can immediately see is that while in the parents the chromosomes are highly condensed, in the hybrid they rather look like more long stretch structures. So indicating indeed there's a change in chromosome structure, there's a chromosome decondensation occurring. And chromosome condensation is connected with DNA methylation. For that reason, we also profiled the mesylome of the endosperm of the parent and the hybrid. And what we see indeed that in the hybrid, this is depicted here in blue using the same color code, red and green are the parents, that on transposable elements in the hybrid endosperm, we have a massive depletion for CEG and CHH methylation, which is how CG methylation is not disturbed. So basically, the chromosome condensation is reduced, and this connects with the reduction in CG and CHH methylation. And with that, I will put things together in a working model. It's not really much more than a model, but this is where we are currently. So pericentromeric regions, as I said, are enriched for transposable elements, and these regions are heterochromatic. And this heterochromatin establishment works very well in the parents. However, as I've showed you, it does not work very well in the hybrids. We have a reduction in DNA methylation depicted here by these lollipops. And what we also assume is that in pericentromeric regions, we have binding sites for transcription factors, for instance, Ferris1 and orthologs. And Ferris1 binding, and I did show you the data, but this is what we published, that Ferris1 binding is repelled by DNA methylation. So Ferris1 would not bind to binding sites in pericentromeric regions because they are methylated. Now in the hybrid endosperm, because of low levels of DNA methylation, potential binding sites become accessible. Ferris or other AGLs can recognize them, activate them, or let's say bind to them and activate potential genes located in those regions, giving rise to the phenotype that we observe. And with that, I would like to put things into a final model, going back to what I started with, explaining that new species arise generally by the accumulation of genetic differences over time that finally cause populations to be reproductively isolated. Polyploidization, as I showed you, is a fast track to this process. So as soon as a species is polyploid, it is reproductively isolated from its non-poly progenitor. And the reason for that are deregulated AGLs, deregulated downstream targets, which are imprinted genes, which then establish a triploid block for reasons that we do not completely understand, but we would like to, of course. And these diploids and tetraploids, they will evolve independently. Because they're reproductively isolated, they will accumulate mutations, and with time they will be also recognized as different species. But the reason that they are reproductively isolated happens way before in response to polyploidization. And a kind of similar mechanism happens in response to selfing. Also, selfing leads to a reduction in AGL expression, I have shown you. It also leads to reduction in PEG expression. I have not shown you that, but this we published a while back. And this probably establishes an incompatibility of a selfing with a non-selfing species, in connection also with changes in chromatin structure that we also do not completely understand how that comes. And this selfing and non-selfing species will also evolve independently. They will accumulate additional genetic differences and with time also look different. But also here, most likely, a prior barrier is established way before in the endosperm. And with that, at the end of my seminar, I have during the talk mentioned those people who played the major role in the data I've shown you. I would just like to mention also Juan Santos Gonzalez, our bioinformatician, and Cecilia Verdick, who has been involved in all projects and helping with all kind of efforts. And with that, yeah, I would like to thank you for your attention, and I'm more than happy to answer any question if you have them.
Thank you, Claudia. Very fascinating talk. I see that we have plenty of questions in the Q&A already, so I will start with this one. So that's a question from Marie Mirose. She's asking if, she said very interesting talk. Did you find Helitrons or other Ts in the capsular genome close to the ferrous targets?
Yeah, very good question. And I have to confess, we have not looked at it intensively, mainly also because Helitrons are not so particularly easy to really pinpoint because they're not very, very well defined. But yeah, we don't know. But a good question.
So the next question is from Bjorn Plotner, asking, so in a population where polyploidization occurred in nature, do both plants, so both the 2N and the 4N variants, coexist?
There are very good examples. There are several examples where diploids and tetraploids coexist, yes. Of course, not in all cases. In particular, also sometimes it's not, yeah, if they still look similar, one would need to measure ploidy to really be sure that you have diploid and tetraploid populations. And there are only few studies who have done that, but there are examples, yes.
So there's a question about the natural variation of the triploid block in Arabidopsis. So is there any evidence and known major causes for variation in the triploid block in natural population in Arabidopsis?
There is natural variation for the triploid block that has been also published 2 years back by Rob Martinson's group, and together with us, one major reason for that is natural variation for one microRNA, mere 145, sorry. But there are certainly also other reasons because this is most likely not the only factor contributing natural variation. But that's a very relevant question. We would also like to understand what is the cause, yes.
So next question from Michael Stam. What is your idea why there is less CHG and CHH at pericentromeric regions? Are CMT3 and CMT2 expressed lower? Or are the H3K9 methyltransferases expressed lower? Or any idea why this would be the case?
Yes, yeah, very good question. The expression of CMT2 and CMT3 is, to my knowledge, not significantly changed, but we can only speculate. So one speculation one could have is, I mean, both species differ also in TE content. So if you assume that remethylation depends on maternally deposited small RNA, as has been proposed, you create an imbalance. If you bring in a paternal genome, which is hypomethylated, and has to be remethylated potentially by maternally stored small RNAs, there will be an imbalance. So there may be somehow insufficient amount of small RNA to guide the remethylation. That's somehow how we speculated could work, but so far it's a speculation.
Thank you. So we have one question here wondering about, so if the ferrous 1 and 2 double mutant is showing any germination phenotype?
Testing germination is usually something you really need to do thoroughly under controlled conditions, and we have not done that. I'm aware there is one publication, I think, claiming that there's a difference, but we have not retested that. At least under our conditions, there is no dramatic, let's say, no obvious phenotype, let's call it like that. But let's say, yeah, there may be a minor effect that we have not spotted.
Okay, and I think we have time for one last question. So actually the question I have. So you said that Ferres 1 and Ferres 2 are very similar. So do you know if there is any redundancy between Ferres 1 and Ferres 2? And if you look in the Ferres 2 single mutant, if there was any obvious phenotype there?
We have not looked at the single mutants, no. Actually, already the double mutant, let's say in the diploid state, we see genes being deregulated, but not a lot. So we believe that there's a higher level of redundancy with other HALs that we try to identify. So for that reason, we have not spent much time looking at the singles. Is that the answer to the question?
Mm-hmm. Yeah, it does.
Okay, good.
Thank you, Claudia. So with that, I think we need to wrap up this first session. Thank you very much, Claudia, for the great talk and the great answers. And I will now move to our second speaker of the day, who is Sunil Kenchanmane Raju. So Sunil is a postdoc at the Department of Plant Biology at Michigan State University in the States. Sunil received his PhD from the University of Nebraska at Lincoln, where he showed the utility of induced epigenetic variation in plant breeding for yield and stability-related trait in soybean and Arabidopsis. His current research interests focus on plant resilience to climate change, which is very timely and important, with a particular focus on understanding how chromatin dynamics and epigenetic variation in a stress-adapted crop wild species can help tackle this question. Sunil is also an active member of the American Society of Plant Biologists. He's serving as a plant cell assistant features editor. And is also currently the outgoing chair of the ASPB Ambassador Alliance. Sunil, it's great to have you here today, and take it away.
Thanks, Matthias. Let me share my screen. Okay. Thanks, Mary and Katie, for the invitation. So as Matthias mentioned, I am a postdoc in the Chad Niederhuth lab in Michigan State University. And today I'll be talking about my recent work in the lab, where we looked at DNA methylation signatures of duplicate gene evolution in angiosperms. And we have recently posted this on the bioRxiv. And it's also an honor to be speaking after Claudia, whose work I have admired for long. And then today I'll be talking about duplicate gene evolution in angiosperms that's related to some of the things she mentioned. So polyploidy or whole genome duplications are more prevalent in plants than any other eukaryotic lineages. So that's why when you look at most of the angiosperms, they would have likely had a polyploidy event in their evolutionary history. Whole genome duplicates and also small scale duplications, which are also called single gene duplications, lead to higher gene content in the organism. And this higher diversity, particularly in the plant genomes, are linked to environmental adaptation and domestication. So as you can see here, angiosperms have gone through multiple whole genome duplications, some specific to angiosperms and some specific to individual families. And one of the prominent ones is the example of a recent duplication in the maize genome. So as I mentioned, there are two major forms of gene duplication. One is the whole genome duplication, and other is the single gene duplications. So in the whole genome duplications, the entire gene content of the organism is copied over. So you can see this as a remnant. The remnants of this in the present day organisms can be seen as syntenic collinear genes. The other forms include the local duplications, the tandem, which where a copy of a gene is copied over close to the original copy with or without intervening sequences. And then there is the non-syntenic duplication forms, where a gene is copied over in a different chromosomal location, maybe within the same chromosome or far away from the chromosome. So the overwhelming majority of the cases, duplicate genes end up being lost. So as you can see here, in most of the cases, they get lost. They can also be conserved, but in some cases, they assume a new function. And there are also cases where they assume subfunctionalization or specialization. And this can happen in two ways. One is at the functional level, and the other is at the expression level, where you see differences in expression in cell types or different tissue types. And multiple hypotheses has been put forward to describe this. One of them is the gene balance hypothesis, which talks about preferential retention of gene duplicates in whole genome duplication. The other one is the epigenetic complementation model put forth by Rodin and Riggs, which suggests that immediately after duplication, you see epigenetic silencing that protects the gene duplicates from accumulating deleterious mutations or pseudogenization. Conforming to this model, Keller and E have found in the human duplicate genes that the younger genes tend to show higher DNA methylation, whereas in the older duplicate genes, you see lower and lower DNA methylation profiles. So as Claudia also mentioned, DNA methylation has an effect on gene expression. So basically, DNA methylation is the covalent modification of the cytosine nucleotide via methyltransferase activity. And in majority of the cases, DNA methylation leads to transcriptional inactivation. In plants, we see DNA methylation in 3 different contexts: the symmetrical CG and the CHG context, as well as the non-symmetrical CHH context. And different enzymes like MET1, CMT3, and CMT2 are responsible for methylating in these 3 different contexts. And different DNA methylation context and the location of DNA methylation dictates how they affect gene expression. For example, in this figure, you can see methylation in the CG context in the transcriptional start site leads to silencing of the gene, whereas the same methylation profile on the gene body and depletion of methylation marks in the transcriptional start site and the termination site leads to higher expression of the gene. And also, if you have higher DNA methylation in all 3 contexts all over the gene, you see RDDM silencing of the gene. So multiple studies have shown relationship between DNA methylation and duplicate gene evolution in individual taxa in plants. However, these studies use different methods to analyze DNA methylation data, as well as they use different criteria to classify genes based on their methylation classification, making it harder to derive broad conclusions from these data. So what we did was we used 43 angiosperms. As you can see, I used Amberella as our out species. We used a bunch of monocots as well as prominent dicots like cotton, important family like Brassicaceae, legumes, as well as Solanaceae. And then we looked at the genes and classified them into different modes of duplication using tools developed by Andrew Patterson's group. And then we took the methylomes from these 43 angiosperms and classified them into classified the genes into genic methylation classifications, into gene body methylation, characterized here by CG methylation on the gene body, transposon-like methylated or TEM genes, characterized by methylation in all 3 contexts all over the gene, as well as unmethylated genes. So first, I was interested to look at the distribution of these genic methylation classifications on the genome, and also wanted to test whether transposon-like methylated genes are particularly enriched in the highly heterochromatic pericentromeric region. As you can see in Arabidopsis, as most of you already know, the TE density is higher in the pericentromeric region, whereas gene density is higher in the chromosomal arms. And corresponding to this, we see higher density of gene body methylated genes shown in cyan here in the chromosome arms, and higher density of TE-like methylated genes shown in golden here in the pericentromeric region, corresponding with higher density of Ts in the pericentromere. But this trend is not uniform across all species. As you can see in banana, even though there is higher concentration of Ts in the pericentromeric region, we don't see higher density of transposon-like methylated genes. And similarly in soybean, we see the same trend. We don't see higher density of transposon-like methylated genes in the pericentromere. So then I looked at all 43 species and looked at how these different genes are distributed across the chromosome. So then when you see here, there is negative correlation of gene body methylated genes and unmethylated genes with the transposon content. But we see a positive correlation for transposon content and TE-like methylated genes in 28 out of 43 species. But we still see some important differences where some species have negative correlation with transposon content and TEM genes. And this was particularly seen in the monocot family as well as the legume family. So then I looked to see if this trend is significant, and I used Pagel's lambda to see if there is a phylogenetic signal to this trend. Pagel's lambda uses a Brownian motion model to look at the trait evolution, and we see significant phylogenetic signal for these trends. And then I also was interested to see if genome size could be a reason driving this relationship. As you all know, transposon content is one of the major forces driving genome size. So when I looked at the genome size of all these 43 species, I do not see any phylogenetic signal. So this suggests that the negative correlation between transposon content and T-like methylated genes that I see here is not primarily due to genome size differences or differences in TE content, but probably because of the differences in distribution of these TE contents. Then I wanted to know between the duplicate genes, how DNA methylation is affecting the divergence and conservation of differences in expression or gene function. So for this, I looked at the enrichment and depletion of different classification of genes with different modes of gene duplication. As you can see here, whole genome duplicates are depleted of transposon-like methylated genes, but they show enrichment for gene body methylated genes. And also for the local duplications, you see tandem and proximal, they show depletion for gene body methylated genes, while they show enrichment, some enrichment for transposon-like methylated genes. But what was interesting is we see that for the transposed duplicates, we see depletion of transposon-like methylated genes. But our original assumption was that because dispersed and transposed duplicates have a duplicate copy in a different chromatin context, we assume that we will see higher TE-like methylated genes in the transposed duplicates. But we see the opposite trend. And later in my talk, I'll show you results that indicate what we think is happening with these trends. And then the next thing we looked was between the two pairs of duplicate genes, do we see a difference in the genic methylation classification, or if they have same DNA methylation profiles? So when we looked at across 43 species, here I'm only showing results from maize, but majority of the genes shown here in blue show same DNA methylation profile among the two duplicate gene pairs. But we see some proportion of genes, particularly in the non-syntenic duplicates, the dispersed and transposed, we see a significant amount of genes that show different DNA methylation profile. For these modes of gene duplication, it's not easy to see which gene is the parental copy and which one is the recently duplicated copy. But for the transposed duplicates, we can ascertain the parental copy as well as the recently duplicated copy. So I leveraged that information for the transposed duplicates. And as you can see, again, I looked at across all the species. You can see that in the parental copy, you see higher proportion of gene body methylated genes, and they reduce in the transposed copies of the duplicates, whereas we see increase in the transposon-like methylated genes in the transposed copy. So what this is showing is that a big proportion of gene body methylated and unmethylated parental genes switch to TE-like methylation in their transposed copy. So this led us to thinking about how DNA methylation may be contributing to the divergence of gene duplicates and the timeline at which this happens. For this, I looked at synonymous substitution rates, KS. So synonymous substitution rates have been used to determine the age of the genes in the gene duplications, because the assumption is that when you have synonymous substitution rates, they usually do not affect the function of the genes. So as the gene ages, they tend to accumulate synonymous substitutions. So as you can see here, if a gene shows lower Ks, it is a younger gene, and then as the gene ages, it accumulates more Ks, and then it's an older duplicate. So here you can see from Arabidopsis whole genome duplicates that the younger whole genome duplicates tend to show higher gene body methylated genes, whereas when you come to single gene duplicates, we see younger duplicates showing higher transposon-like methylation. So this trend is similar when we look across different modes of single gene duplications. Here, again, I'm showing whole genome duplication as well as a combined plot for single gene duplicates, and also separating them based on local duplications or based on the non-syntenic duplication. So as you can see here, in all the single gene duplicates, we see enrichment of a transposon-like methylation in the younger duplicates as opposed to the older duplicates. I also used another independent method using MC-ScanX transposed, which differentiates different transposed duplicates into epochs based on speciation events. So even those results confirm and support this idea that the younger transposed duplicates have higher TE-like methylated genes, whereas the older genes show more gene body methylated genes. So then I re-examined the distribution of these Ks, the synonymous substitution rates, in all genes across all species, and then plotted them based on their median Ks values. So this was really exciting to see that the species which have a higher proportion of younger transposed genes, shown here with a lower Ks values on the y-axis. So these species showed enrichment for transposon-like methylated genes. Whereas when you look at the species which have a higher proportion of older transposed duplicates, they are depleted in TE-like methylated genes. So this now goes back to this goes back and explains why we see a depletion of TE-like methylated genes in the transposed duplicates. Again, if you look closely here, there are some genomes which show enrichment of TE-like methylated genes. And these are, for example, maize and soybean and other genomes which have a higher proportion of younger transposed genes. So this goes back and explains the reason why we see a depletion in most of the species which have higher proportion of older transposed duplicates, as opposed to an enrichment of transposon-like methylated genes in the transposed duplicate copies. So then I was also interested to look at how selection acts on different modes of a different classification of these genes, and also on the different modes of gene duplication. I used Ka-Ks ratios, which is the ratio of non-synonymous substitution rate with the synonymous substitution rates. A Ka-Ks ratio of 1 indicates neutral selection, whereas a Ka-Ks ratio of greater than 1 suggests positive selection. As you can see from this figure, most of the genes showed Ka-Ks ratio closer to 0. That means that they are under purifying selection. Also, it says that in these genes, non-synonymous mutations are not tolerated. But if you look at this, the plot for the single gene duplicates, you see that the transposon-like methylated genes, they show higher proportion of genes show an increase in Ka-Ks ratios. And a small proportion of them also have Ka-Ks higher than 1. So when I look at genes which have Ka-Ks ratios of greater than 1, I see an enrichment for transposon-like methylated genes and a depletion of gene body methylated or unmethylated genes in those species. So this suggests that transposon-like methylated genes might be under relaxed selection. And also I looked at presence absence variation genes in different genomes, and transposon-like methylated genes are enriched in those presence absence variation genes, again suggesting that these genes could be under relaxed selection and might be contributing to the divergence. So all the results I presented here are showing interspecies variation across 43 angiosperms. I also looked at within species variation using Arabidopsis thaliana. So I took 928 methylomes from different Arabidopsis thaliana accessions and looked at how different methylation classification and modes of duplication affect them. And I see very interesting trends in the frequency of epialleles in the 928 accessions, and they were different based on the mode of gene duplication as well as the classification of the genes. I do not have time to go through those results, but those results are in my preprint. So finally, to summarize, what we see is there is lineage specific difference in the transposon-like methylated genes and the gene distribution, particularly in the monocots as well as the legume families, and they show a strong phylogenetic signal. And also, irrespective of the mode of gene duplication, we see that majority of the genes retain same methylation profile, and a small proportion of the non-syntenic duplicate pairs show changes in duplication status, and mostly they are showing higher transposon-like methylated genes in the recent duplicate copy. And one of the very interesting results we got is that the younger single gene duplicates tend to be enriched in transposon-like methylated genes, while younger whole genome duplicates tend to be enriched in gene body methylation. Again, this confirms to the gene balance hypothesis. The next interesting result we saw was that when you look at these transposon-like methylated genes, they tend to show a higher KAKS values across different species. And then a proportion of them even show KAKS values greater than 1. So this, I think these results totally suggest that what we see here is an extension of the expression reduction model, where you see that the duplicate genes acquire higher DNA methylation to protect from pseudogenization, but also to maintain the balance initially. But our results hint that we see these genes are also enriched for those which have higher KAKS values, suggesting that transposon-like methylated genes might be helping in the divergence of these gene duplicates. With this, I would like to thank my advisor, Chad Niederhuth, for his help with introducing me to this project and helping me with the analysis. I would also like to thank Marshall, who was an NSF RU student in our lab and who did some of the initial analysis. I'd like to thank Pat Edger, Rob Van Buren, and James Schnabel for their comments on the earlier version of the manuscript, and also my lab mates, Eleanor Ritter and Hannah Brown. And I just want to say that we have the preprint submitted, and if you have any questions, you can contact me on my email or through Twitter. And with that, I'll be happy to take some questions.
Thank you very much, Sunil, for a great talk, very fascinating data. We have already plenty of questions in the Q&A box, and I will start with this one from Andrew Fowdry. He's asking, do you believe the patterns for methylation of young single gene duplication events will also be seen in transgenic genes or transgenic events?
Yes, so that is like an extension of this work. So I think when you have multiple copies of a gene, you might have them in a different chromatin context, or you might have it in the same chromatin context as the original gene. And this might be important in looking at how, this might be important in looking at how transgenics, like introducing a gene, might affect in the genome, how methylation might act on these transgenic genes. So yeah, that's a big possibility that it could be one day used in those studies.
Maybe a follow-up question would be, do you think that the duplicated copy would have the possibility or the potential to also influence the chromatin state at the receiving site?
Can you ask that question again? Sorry.
So do you think that the insertion or the presence of the duplicated copy at a new chromatin location would influence the chromatin state at this receiving location?
Yeah, so I don't know if that is so prominent, but like Claudia was mentioning, when you have TEs that move on to a different location, they influence the chromatin architecture or DNA methylation, particularly of the neighboring genes. But having a gene copy moved, I'm not sure there are examples to show that those chromatin or DNA methylation profiles spill over to the adjacent genes. But for sure, in the case of transposon, it happens.
That's good, thank you. The next question is from Chandra Misra, asking, does the genic methylation across species in chromosome arms versus pericentromic regions is tissue dependent, as it can be seen through methylation in reproductive tissues, by example?
Yes. So in the next iteration of this work, in the further studies, what we are trying to look is, because transposon-like methylated genes, some of them, when we have carefully examined these genes, we see that they have different expression patterns in particular tissue types, particularly when you see reproductive tissues or in the endosperm or in different developmental stages. So we think that these genes might be mostly silenced in majority of the developmental stages, but they show expression during specific stages. So then it would be interesting to see if we have transcriptome data for multiple tissue types and multiple developmental stages. We'd like to see how these transposon-like methylated genes behave under those conditions and when they are expressed, and is that the reason why they are suppressed in majority of the tissues, and then they have I mean, for example, in reproduction, we know that there is demethylation, and maybe that's the reason why they're gaining expression there, because they have a prominent role there. So yes, we are trying to look at those for species where we have comprehensive transcriptome data, for example, Arabidopsis, maize, and rice. We'd be looking at those and trying to see if there is a correlation between specific expression patterns.
Mm-hmm. Sounds good, thank you. And I think we have time for one last question, and it's about your transposed, your duplicate copies. And so further than DNA methylation, have you got, or are there around data about other chromatin marks such as F3K9 demethylation, or maybe presence of small RNAs in those transposed copies?
Yes, so there are, I think there is one study that looked at other chromatin marks than DNA methylation in the duplicate genes, and they looked at how they might be affecting expression patterns in Arabidopsis as well as expression patterns and evolution. But we have not looked into other chromatin marks, and in the future it'll be interesting to see how those histone modifications might be affecting evolution of these genes. But yeah, we have not looked into those right now.
Great. Thank you very much. So that was the last question for you, Sunil. So I would like to thank you, Sunil, and thank Claudia again for great, exciting seminars and a really nice discussion during the Q&A session. And now I will hand it over to Katie for the conclusion.
Yes, I'd also like to thank you all for attending, and thank you to our speakers and Matthias for doing a great job with the moderation. Thank you all for sending questions. There was a long list of them, so feel free to continue the conversation over on the Plantae blog or on Twitter. We'd love to hear from you and hope to see you in our next webinar. Until then, hope you have a great day or evening, depending on where you're at. Thank you.